So hopefully a lot of you were here last week, and if you weren't, welcome. And so this is the Modern Research Techniques series. This is sponsored by a training grant from the National Institute of Health that's allowed us to collaborate, so the University of Rochester Medical Center to collaborate with Roberts. And so as part of this series, we are bringing graduate students and postdocs here to Roberts to get teaching experience and to talk about some modern research techniques, and hopefully you guys in return get to learn about some cool stuff that's done in biomedical research. So as you came in, hopefully you signed in. If you didn't sign in, if you could sign in before you leave. And also hopefully you grabbed 2 evaluation sheets. If you would evaluate both Sarah and Monica, that would be great, especially faculty and staff. If you're here, we'd like you to evaluate them as well. So each presentation is going to be 20 to 25 minutes. If you have questions, feel free to interrupt. So feel free to raise your hand or stop Sarah or Monica if you have a question, or there'll be time after each presentation for questions too. So whenever you're most comfortable, but please feel free to ask questions. So to get started, I'm going to introduce Sarah Dickinson. So Sarah Dickinson is a PhD candidate in toxicology, and she's also a Roberts graduate. So she graduated from the biology program in 2014. And today, Sarah is going to be talking about RNA-seq.
All right, thank you, Morgan. Can everybody hear me? I'm usually good at projecting, so just like wave emphatically if you can't hear me. So can I just get a show of hands of who here is either a bio major or has taken bio lab before? Okay, nice, a lot of you. So when I was here, I don't know if you guys still do this, but when I was here, for one of our labs, we got to go to Black Creek Park and dig through the ponds and look for stuff. And while I was also here, I learned about planaria, and these are my favorite little microorganisms. If you guys have never gotten the chance to learn about them, you should. But I want everybody to kind of imagine for a moment that you are on a trip to Black Creek Park, and you are interested in researching planaria. So you go out to the park, you sift through the water, and you get some samples. And you make an interesting discovery. You find 2 different populations of planaria. One of them is the normal, everyday, run-of-the-mill planaria, and the other one is a little bit different. It's actually translucent, and you can see all of its vessels and all of its organs and everything in there. So this is a hypothetical situation. I do not know if these planaria exist, but being all the curious scientists that you are, you decide to research this a little bit. And you all come to the hypothesis that maybe the difference between the 2 populations of planaria is that the translucent planaria aren't expressing the genes necessary for skin pigmentation. So I just want to ask a general question. It's okay if you don't know the answer. When we're thinking about gene expression, what aspect of biology do you guys think we would want to research and look into? I won't let the awkward silence go for long, don't worry. Okay, a follow-up question. How many of you guys, if I say the central dogma of biology, how many of you guys know what that means? Okay, a decent show of hands. So for those of you who don't know, the central dogma of biology, boiled down, says that DNA makes RNA and RNA makes protein. So most of us are familiar with DNA in some context. We know that DNA is what eventually allows us to make the proteins that our body needs to survive. And the central dogma shows that you don't just jump right from DNA to protein. There's actually this intermediary step called RNA. RNA in some ways is very similar to DNA in the sense that it's a nucleic acid, but there's a few minor differences such as base differences and strandedness. And what's really interesting about RNA is really reflective of what genes are being actively transcribed. So if an organism needs a particular protein to be built, either because it's in that stage of life or because of something in its environment, the RNA levels will show what genes are being actively transcribed. And so in research, there's a lot of different ways that you can go about studying RNA. The technique that we're going to talk today about is RNA sequencing. So this is just a general overview of RNA sequencing. I'm going to go into it a little bit in depth. But you're going to start with RNA samples from 2 different populations. So if we were to go back to our hypothetical situation, we would have our normal planaria and we would have our translucent planaria. And we would take samples from both of those and convert that to cDNA, which we'll talk about a little bit more. And then we're going to do something called sequence by synthesis, which is another name for RNA-seq, where you're actually going to identify what nucleotides are in the RNA strands by synthesizing a new strand that's complementary to that. And then what you can do is you can go back and play basically a matching game where you take the RNA that you sequenced and match it back to the genome that you have for the planaria. The end goal of this is to identify the presence and the quantity of RNA transcripts that you're interested in. So the overview of the steps, this slide is very boring and dull on purpose to drive home the point that the overview and the general steps is pretty simple. You prep your samples, you do the sequence, and then you analyze the data. But RNA sequencing is a very in-depth and complicated process, and so all of these steps have lots of different steps and parts to it. And so I'm going to go over them kind of in-depth and kind of not in-depth, but we'll talk about it a little bit. So to start off, you need to isolate your RNA. This is pretty simple because there are a lot of, in the lab, there's a lot of kits that you can buy that will come with different columns where you can just apply your sample and then through various washes, get your sample out. When we're talking about gene expression, we're mostly interested in the type of RNA called messenger RNA, because that's the main transcript that you get after DNA transcription. And that's most reflective of gene expression. What's interesting to note is that you have lots of different types of RNA in your bodies. One of the main types is actually ribosomal RNA. And so in an experiment like this, what you'll often do is you'll actually get rid of that ribosomal RNA in your sample, so you only have the messenger RNA. Once you get your RNA out, you need to check the quantity and the quality just to make sure you've got good quality RNA. And this can be done, they have pretty simple UV-Vis equipment where you can just drop a little part of your sample on it, and it'll measure the concentration and the quality for you, which is very convenient. So after you get your RNA, you need to break it up into small pieces. This step is necessary because the instrument that you're going to use for RNA sequencing can only sequence nucleotides that are about 40 to 400 base pairs long, and RNA sequences can be upwards of thousands of base pairs. So there's a fragmentation step. It's pretty easy. You're just going to either use enzymes, heat, or a sonication method to break it up into small pieces. So afterwards, we want to convert our RNA to cDNA. So the C in cDNA stands for converted DNA. You're basically using RNA to make DNA. There's a couple different reasons why we do this. One of the main reasons is that RNA is really unstable. So RNA, if it's not properly frozen and protected, will degrade very quickly, whereas DNA is a much more stable nucleic acid. So by doing this conversion, we're sort of preserving the integrity of our samples. We use polymerase chain reaction, which stands for, or you can simplify as PCR, to do this conversion. PCR is used in a lot of different techniques, and it's actually used multiple times throughout RNA sequencing. But the main overview of the process is that you use cycles of heating and cooling to separate out your strands, allow DNA polymerase to synthesize a complementary strand, and then reannealing. So you start with a very small amount of RNA, and you end this process with a large amount of cDNA. And that cDNA is what we're going to use moving forward. Once you get that cDNA, you're going to add what are called adapter sequences to the ends of the DNA. And what that allows for two things. The first thing that it allows the instrument that you're going to use to sequence your samples to actually recognize and sequence the samples that you have, which we'll talk about in a little while. The other thing is that the adapters can also act as barcodes. So if you have a variety of different samples that you want to sequence, and you want to be able to go back and see which sample belongs to which, you can use different adapter sequence pairs for different samples. And then as long as you know what you added, you can go back later and identify what you had. So after you add those adapter sequences, you do another PCR step. It's the same kind of concept as before, except now instead of making cDNA from RNA, we're just amplifying those sequences with adapters on the ends. So the next step is we get to sequence the library. So the first part of this is what's called cluster generation. So this image right here is what's called a flow cell. This is what you actually stick into the RNA sequencing instrument to actually do the sequencing. So the cell has a bunch of glass lanes shown here, and on those lanes you're actually going to coat them with a lawn of oligonucleotides, which are just small sequences of nucleotides. Those nucleotides that you're putting on the glass slide are complementary to those adapter sequences that you added to your DNA previously. And so once the lawn is fully coated, you can actually apply your DNA to it, and the DNA will hybridize to those complementary pairs. So starting up here, we have, this is an example of a DNA sample bound to a complementary nucleotide. And what happens is that DNA is going to fold over and form a bridge structure, and then you can, then you'll get DNA amplification of that strand, so you'll then end up with two strands. And the reason for this is just, since we have this big lawn coated with oligonucleotides, we want to make sure that the lawn, before we do the sequence, the lawn is filled with DNA strands. So you're going to go through that strand synthesis cycle, continually. It's just showing one here just for visual simplicity, but across the lawn you have hundreds of thousands of these occurring simultaneously. And so at the end what you're going to do is you're going to cleave these bridges and you're going to be able to move forward with the sequencing. So this next step, the sequencing portion, this is where it gets its name, sequencing by synthesis. So we're doing what we've done a lot throughout this process already. We're going to synthesize a complementary strand to our DNA. But this step is a little bit different. We still have our DNA polymerases and primers that are always essential for this kind of thing. And the nucleotides that are going to be added are a little bit special. So these nucleotides have fluorescent probes attached to them. And any time the DNA polymerase adds one of the nucleotides, it's going to cleave those fluorescent probes and you get a flash of light. And the instrument that you're using can actually image this and it'll associate a particular color with a particular nucleotide. So you'll start at the top and you'll add nucleotides sequentially that are complementary to the strand that you're interested in. And every time a nucleotide is added, a flash of light happens. So this is occurring very quickly. And so this is just a snapshot of what the instrument is seeing at one point in time. So this is an image of one part of one of those flow cell lanes. This isn't even the whole thing. And you can see there's billions of these little lights happening simultaneously. And so as it's going, the computer is constantly recording and imaging these things, and then it can backtrack later and figure out what nucleotide was added to what strand. And so you're essentially sequencing the strand by synthesizing a new one, which is how it gets its name. So after you go through the experiment, there's the data analysis step. I'm just going to talk about some of the theory of this, because a lot of this is just you run a program and put your data into the program, and the computer program tells you how to do it. I don't know if there's any computer science majors here, but I'm sorry. I do not fully understand all the computer science behind it. But the data that you're getting, if I go back here, there's billions of these things. And so the data that you are getting is, can be upwards of like gigabytes per experiment. So it's a lot of data the computer's got to be able to handle. So on the left, we have the raw data. I know it's super interesting to look at these sequences of random things. I'm not going to go into too much depth. The second line shown here, that's the actual sequence that the instrument detected. The bottom line are quality scores that are generated for each of those individual sequences that are added. So over here looking at B, this is a more kind of user-friendly way to look at quality scores. So on the x-axis, we have the position of the nucleotide for a particular group that's being looked at. On the y-axis, we have a number scale from 0 to 40. That stands for the quality scale. And so ideally, any sample that you're going to look at, you want a quality scale or a quality score of anywhere from 30 to 40. So that's shown in the green. You can sort of get away with samples that are in the yellow, which would be anywhere from like 20 to 30, but you really want to avoid looking at any samples that are in the red. So because this is such a kind of expansive and in-depth technique, there are a lot of different things that could contribute to good quality scores or bad. I'm just going to give you two examples. So this is, again, kind of like that snapshot that I was showing you where a nucleotide is being added and you get that fluorescent light being shown. So this is just one snapshot. Sometimes what happens when a nucleotide is added is that you get that probe cleavage, but it's kind of a dull color shown here. And with that dull color, the instrument isn't really super confident that the nucleotide it's saying is added is the correct one. And so this would get a low quality score. Another issue that can happen is called low diversity. This happens when you have fragments of your sample that are all near each other and that have the same nucleotide being added at the same time. So you get the same color in the same general area. And what happens with this is sometimes the colors can actually blur together and the instrument isn't sure anymore where the fragments are on the flow cell. This can be a particularly big problem early on in the experiment when the computer is first trying to figure out where are all of the DNA that are bound to the sample. So once we have our reads all finished, we need to align the reads to a genome. In our particular hypothetical situation with the planaria, I think, don't quote me on this, but I think we pretty much know the whole genome of planaria. And so what we would do is we would go onto the computer and find the genomic sequences of planaria, and then we would have the computer break them up into little pieces. And then we would also have the computer match the fragments that we measured in our samples to places on the genome. The reason that we have to fragment up the genome is because if you look around the room, all of us have human DNA, but all of us have very different expressions of that DNA. And so even though we all share human DNA, there are variations between our DNA. And it's the same thing with organisms in the environment too. While they all share the same basic DNA, there's different things like single nucleotide polymorphisms that differ. And so if you have a lot of those in a fragment, it might actually cause a mismatch when you're looking at the genome. And so by fragmenting it up, you basically increase the probability that you'll get correct matches. So with the data analysis, there's a lot of it that goes into it. And I just have two kind of scenarios of things that you need to keep in mind when you're doing data analysis. So looking at A, we're comparing gene A in two different conditions. So this could be one gene in our normal planaria versus one gene in our translucent planaria. So we have the number of reads. For condition 1 in gene A, it's 20. And for condition 2 in gene A, it's only 10. So if we just look at the number of reads, it looks like gene A is being expressed in condition 1 twice as much as condition 2. However, you also need to factor in the total number of mapped reads. So when you're thinking about mapped reads, these are the reads that actually get mapped to, for lack of a better word, an important part of the genome. So on your gene, you have portions of the gene that actually code for protein. And for when we're looking at gene expression, that's what we're mostly interested in. So you may get a lot of reads to the genome, but we need to make sure that the reads that we're focusing on are the ones that actually code for protein. So when we looked at the total number of mapped reads, condition 1 has 20 million mapped reads, and condition 2 only has 10 million. So when you divide, their expression values are actually the same across the board. Looking at B, this is looking at two different genes now, and these two different genes are of different lengths. So gene A is 4 times as long as gene B. Now, if you look at just the reads, gene A gave 20 reads, whereas gene B gave 10 reads. However, you have to factor in the size of the gene that you're looking at too. So when you divide them by their lengths, you see that gene B, despite having less reads, was expressed twice as much as gene A. And so this equation here, you don't need to pay too much attention. This is just an example of an equation that some of the software will use to factor in both mapped reads and the length of a gene. So looking at numbers and tables isn't all that user-friendly. So this is an example of two ways that you may present your RNA sequencing data. So over here we have what's known as a volcano plot. So on here, the authors of this paper actually plotted every single gene that they sequenced. And they separated them based on the fold change of expression compared to their control. So on the bottom, they have the log fold change range starting in the middle at 0 and then going to the negative side on the left and positive on the right. Positive indicating that there was an increased expression level and negative indicating that there was a reduced expression level. Then they highlighted in red the values that were statistically significant. So those are the ones that were either significantly expressed in a higher manner or significantly reduced in their expression. Now this is kind of useful to visualize the whole gross data altogether, but it's not super useful for looking at exactly what's going on. So heat maps can also be used to kind of examine what's going on. I liked what the authors did on here because on this axis, sorry it's a little blurry presented up, but they actually grouped the genes that they're interested based on what they had in common. So going back to our planaria example, if we found that a group of different genes were differentially expressed, we can look and say, okay, which of these genes relate to pigmentation in planaria? And we can group them and see how many of them are differentially expressed. So on this side, they actually have all of the genes that they're interested in for this figure listed out. And then the colors are what correlates to variations in expression. So the blue color indicates that expression was reduced when compared to their control. And then the red color indicates that it was more highly expressed when compared to control. Okay. So just for the last part, I want to talk about some of the advantages and 1 or 2 disadvantages of RNA-seq. So you'll probably, this might look familiar. This was the first figure I kind of showed showing the overview of the RNA sequencing. RNA sequencing is relatively new. It was first, I guess, invented, you could say, I think around 2008. And one of the biggest things that's starting to replace are microarray analysis. So the microarray analysis shown on this side starts out very similarly. So you start out with RNA samples and you convert to cDNA just like we did in RNA sequencing. But what you're doing here is that you're actually tagging that cDNA with fluorescent probes. You then add your cDNA to a plate, which has a bunch of different wells, and in those wells you have different transcript probes. And you know the sequence of those transcript probes, those are specific for whatever genes you're looking at. And so if your sample hybridizes with one of those probes, you get fluorescence and you know that the gene you're interested in is expressed in that sample. Now, so this is a pretty useful technique. One of the reasons that RNA sequencing is sort of replacing it is because RNA sequencing is much more sensitive and you can measure many more RNA transcripts than you can with microarrays. Another thing is it's how it gives you the data. So with microarrays, you're really looking at the relative intensity of light and equating it to expression level. Whereas here in RNA sequencing, you're actually directly sequencing and adding nucleotides specifically, so it becomes much more of a quantitative and specific analysis. One thing that I didn't talk about much, just because it's a little bit more complicated, but with RNA sequencing, I mentioned earlier that you will map your samples to a reference genome, so you know where the genes came from. You don't actually, if you don't know the genome of your sample, you can actually use RNA sequencing to identify like new genes or to actually sequence. So it's a really powerful technique. One of the biggest downsides is that it's super, super expensive. One of my lab members did an RNA-seq experiment with just like, I think 10 samples, and it cost over $8,000. So it's a very expensive technique. So depending on what you're interested in, microarrays still may be the way to go. So these are references if anybody is interested. And with that, I can take any questions if you guys have any. Okay. That just means I was super clear and you guys all understand RNA sequencing perfectly. Yes. I'm just kidding.